All right, so Professor Ohta.
Thanks for the introduction, and thanks everyone for coming to this seminar virtually. Uh, I hope you have your own pizza or something to enjoy. So I'm going to be talking about research in my lab, and that's the Microdevices and Microfluidics lab in the EE department. Although we do a lot of interdisciplinary work as well. Also, because this is a presentation uh, to everyone in the College of Engineering, I I'm trying to highlight more of the student work um, as well, because many of you will know the people that have been doing the research that I'm going to be talking about in this presentation. So our lab does a lot of work in microsystems. So these are um, microfabricated systems because we are working with small objects such as individual cells that would be in your body. So because of that, we encounter a lot of microscale and nanoscale physics in the work that we were doing. So I just wanted to quickly go over that. And for those of you who are not familiar with it, if you are familiar with it, then you can kind of uh, skip over this part. But what is non-intuitive for most people when talking about forces in the microscale and nanoscale is that the kinds of forces that we experience in everyday life that depend on mass become much less significant in the microscale. And so, things like momentum and other forces, gravitational force, for example, aren't really significant in the scales that we're working at. And other forces, such as surface forces, electrostatic forces, those become much more significant. And this photo here is an illustration of that. So, when you go to the swimming pool or to the beach, you jump in the water, you can get out of the water easily. But if you're a small insect, then the surface tension of the water becomes relatively stronger, and it can present a big obstacle to getting in and out of water. We do a lot of projects that actually take advantage of surface tension because of the fact that it's a stronger force, relatively stronger force in the microscale and nanoscale. So in this talk today, I'm going to be talking about a few projects that we have been working on in our lab. One that has to do with micromanipulation. In particular, we are doing some manipulation of single cells. We also have created microfluidic systems for analyzing single cells, and I'll talk about that as well. We also have started to move in towards some areas that can be considered artificial intelligence or machine learning, but also for looking at analyzing small objects, and for that we're looking at analyzing the health of embryos. And then I'll talk about another area that we have multiple projects in, where we use room temperature liquid metals for various types of electronic circuits. So to start off, this is a partial list of all the people that I am working with on the things I'm describing here. So I have a big list of collaborators. The names that are bolded I'm working on them with projects that are described in this presentation. Many people from University of Hawaii. Um, Actually, and sorry if I'm missing names. I just noticed I'm missing Professor Lloyd Hihara's name there. He's a collaborator on one of the things that we're doing with liquid metal. Also, I have other collaborators here at UH that aren't on here. you know, for example, Joe Brown, who was leading our effort to bring in some clean room equipment. You know, his name's not on here. So this is not a complete list. Sorry if you're not there. As well as all the faculty advisors for VIP teams. They are doing a lot of work as well. And I didn't put everyone's name there. In the green box, these are the current graduate students that I am either the advisor or that I work with. And on the right side, these are graduate students, again, that I've either am the lead advisor or co-advisor for. And the names that are bolded are people that are contributed to the work in this presentation. This top picture is pre-COVID, so hopefully we can have more lab get-togethers like that in the near future. So I'm first going to talk about our work to do micromanipulation and also microsurgery on cells. And this is using a platform that uses microbubbles that we can control using light. This video that's playing on the left side is kind of showing some example of that. So you can see things moving around. We're doing that by using lasers to control the objects in that field of view. This is under a microscope. And all of these objects are on the order of the width of a human hair or smaller, because that's the kinds of scales that you need to work with if you want to work with mammalian cells. The names and photos here are students that have worked on this part of our research. And as you can see, you know, there's several PhD students that have worked on this, including Ashley, who's not an electrical engineering student, but a mechanical engineering student, as well as undergraduate students over on in the bottom right, and that's kind of the year they graduated. Arif is currently a postdoc here at UH, and Julian and Kelly are working locally. Wenchi is now a researcher at Max Planck Institute for Intelligent Systems in Germany, and Ashley is now a faculty member at a university in Beijing. So for this area, we are using small structures to do micromanipulation. And this type of manipulation using these small structures, a lot of times these small structures are described as microrobots. And these are just some examples of various kinds of microrobots that currently exist. A lot of them are controlled using magnetic fields. So that would be these examples, or many of these examples on this slide. Although you can also control these structures using electrical fields, such as the one in the top right. And, you know, in addition to magnetic fields and electrical fields, there's also optical fields. And there are a few examples of using optical fields to control these microrobots as well. But for all of these micromanipulators, these are some of the areas that are being currently researched. One is to increase throughput, or to increase the rate at which you can manipulate objects. Then also to increase the resolution, or what's the smallest object you can manipulate. If you can manipulate them in either 2D or 3D, that is good. Making the control systems work really well. And also, certain kinds of systems will have additional capabilities besides just being able to move around objects. So what we have focused on to contribute to this is working on increasing the throughput of these micromanipulation systems. And we do that by moving multiple actuators in parallel and also moving them cooperatively. And we have also developed a system that has additional capabilities. So for example, we can do molecular delivery to specific single cells, and we can also lyse the cells or break those cells if we desire. So this is the system or platform that we're using to do all of that. And this is a micromanipulator that's optically controlled. So what it is it's a small chamber that's filled with liquid. And inside of that chamber, we create microbubbles using lasers. And we then also use the laser to, once we create the microbubble, to move around those microbubbles and use the microbubbles to manipulate small objects in that chamber. So you can see on this image on the right that says UH. The UH is made out of very small plastic beads, about 20 microns in diameter, so about the same size as a white blood cell. You can also see a video in the lower right of a bubble pushing around a larger object. And there are biomedical applications for this type of manipulation. So we can use this to isolate single cells and do experiments on single cells. You can also position single cells as kind of seed materials for creating artificial tissues and organs as well. So the way that this is working, this is kind of a high level explanation, but we are shining the laser onto a surface that absorbs light. We use either amorphous silicon or a thin film of titanium. When the light shines on that material absorbs the light and it creates a localized hotspot. That localized hotspot creates a temperature gradient, and we use that to temperature gradient to drive some processes that cause the bubble to move towards that hotspot. So we can, wherever we move the light, the bubble follows, and we can use that to assemble objects. So here's an example where we're assembling these hydrogel blocks. So these are blocks of a material that's kind of like Jello, and you can use that to grow cells in 3D. And we're assembling blocks of this material that contain some cells in them into various patterns. In this experiment, we used yeast cells because they multiply very quickly. So we could see the growth of the cells in a very short amount of time. And you can kind of see that in this series of images. The last image on the bottom right shows a lot of cells because they've multiplied quite quickly. We can also use the same kind of platform to move around single cells, so cells that are not embedded in a hydrogel. In this case, these cells are fibroblasts. So these are cells that would be found in your body in connective tissues inside of your body. And as you can see from the video, we are able to, we have a high enough resolution to move around a single cell and assemble them into various locations. So those previous videos showed, one or two micromanipulators working at once. More recently, we have also increased the number of bubbles that we can control at any given time. So this is one of Arif's major contribution to this project. And we do that by using some holograms to alter the wavefront of a single laser beam. So it creates multiple spots at the focal plane of the surface that we're projecting the light on. And so we can create many more bubbles, and each one of these bubbles is individually controlled. And we can then move them around without having any motion dependent upon one another. So this is one of our unique capabilities compared to other micromanipulation systems, such as a magnetic field. Magnetic field, it's very hard to localize. So you can move around many objects in a magnetic field, but they're all going to move according to a more global magnetic field rather than a more localized magnetic field. But here we can move around each one of these bubbles almost independently of the motion of the other bubbles around it. So this allows us to do things like move the bubbles in a cooperative fashion to accomplish a particular task. So the video that you're seeing here, that little square is a structure made out of a polymer. It's hard to move around that square with just a single bubble, but as you can see, adding a second bubble and moving it cooperatively along with the first bubble allows us to reliably control the movement and also the rotation of that object as well. In that same platform, in addition to moving around the objects, we can also perform surgery on the cells. And that is just done by using the same exact fluid chamber. All we got to do is change the parameters of the laser a little bit. So we start pulsing the laser. That causes some fluid streams to form around the bubble that are strong enough to temporarily open some small holes in the cell membrane, and that allows us to deliver molecules into the cell. If you do this right, this is reversible. So the cell will open up some small holes in its membrane. Some molecules can diffuse into the cell, and you haven't bothered the cell that much. So it will reseal the holes and it will still remain alive. So that's demonstrated in this experiment where we delivered a small strand of DNA into the interior of a single cell within this sample. That DNA contains some instructions to tell the cell to start producing this protein that makes it glow under UV light. And so you can see that in the second column of images, you can see on day one, we injected this into one cell. So you see one cell glowing under the UV light. By the second day, that cell has divided. That DNA goes to the second cell as well. And so you can see two cells glowing under that UV light. So we can deliver these small molecules to very specific cells using the same platform. We can also do cell lysis. So that's kind of the same process, except more destructive. So, you know, we're opening small holes in the membrane. If you add more power, you can just open really big holes in the membrane, big enough until eventually the membrane will break. And now the good thing is, well, the bad thing is the cell's dead. The good thing is that now you have access to the materials inside of that cell in case you want to do some genetic analysis or other kinds of measurements on what was going on with the cell. So this is in the same, again, this is the same platform as the manipulation. So we can do manipulation and then we can do lysis on particular single cells, depending upon which cell you want to do some analysis on. So this is kind of our work in manipulation and microsurgery. And you can see that we are focusing on accessing and interacting with single cells. We can also make other systems as well that will work with analyzing single cells. And I'll talk about a couple of those projects in this section briefly. So the students that have been working on this are listed on this slide. So Mohsen, Richie, and Meenakshi are current students right now. And Nalu has graduated. He's now at Blue Origin, the Amazon rocket company. And then you can see Arif's name here again. For one of these projects, we're also working with Ian McPherson, who is in JABSOM. So this is the project that we're working with Ian McPherson at JABSOM on. What they are interested in is measuring the proteins of single cells as a diagnostic tool. But when they measure the proteins of the cells, they want to be able to keep track of which cells they were measuring. So before they do the protein measurement, they want to attach a strand of DNA to these cells that acts like a barcode, like the barcode that are on groceries when you go to the supermarket. And you know, when you check out, that barcode tells the cash register, what item you're buying. So these DNA barcodes would be there to tell us what particular cell we're analyzing. So how do we associate one of these DNA barcodes with a cell? Well, we can do that by encapsulating a barcode along with a single cell inside a tiny droplet. And we do that by making this microfluidic device. You can see the overall device in the bottom left here, where we can inject some cells into here. We inject some DNA barcodes into a different inlet. And then we also inject some oil into this as well to break up the cell sample into discrete droplets. And we time everything so that each droplet will contain a single cell, and that will be added to a barcode. And then the cells coming out of this, we then give to Dr. McPherson's lab, and they do analysis on those labeled cells. Along those lines, we also wanted to look at, okay, can we tell what's going on with single cells without having to add DNA barcodes? And since we're in the electrical engineering department, we decided to see if we can measure the electrical properties of cells and have that tell us something about what's going on with the cells. So these first experiments that we did were with brine shrimp eggs. So that's what you see here. And when you put these, you can get, dried brine shrimp eggs because they have a hard shell around them, and you put them in water, and then eventually they'll hatch. So these dried up eggs start to hydrate, and then eventually the brine shrimp comes out of the egg. So we put these into another microfluidic platform that had some microelectrodes that go near the brine shrimp egg, and we measure their electrical impedance over time. And the point of this was to try to get some correlation between the electrical impedance and what was going on with that brine shrimp egg. So we're able to distinguish between those eggs that end up hatching and the eggs that don't hatch by measuring the impedance and looking at a particular part of the impedance as a function of time. So currently we're trying to expand this to embryos that are a little bit more, or set eggs or embryos that are a little bit more like a mammalian type of embryo, but we're still working on that. So eventually what we want to do is be able to take some electrical measurements and predict if the embryo is going to be or going to result in a birth or not. But this is a little bit more long term. On the shorter term, we have been working with Dr. Thomas Huang, who is also a JABSOM faculty member, and he does work in vitro fertilization. And this is leading into our project that is using machine learning. So what we're trying to do is analyze embryos and use that to correlate embryo health as a prediction for in vitro fertilization. This is one of our newer projects. The lead on this is Yusuf Harun. He's recently graduated last semester. He's now at a PhD program at Rochester Institute of Technology. Meenakshi is a current student. She's taken over on that project for him. Arif, you can see him again. He also helped with some of the early work on this project. And we also have a bunch of undergraduate students working on this project. Two of them were co-authors on some papers with us. That's Josh Mellinger. He just graduated in May. Willie Chang is still around. He's currently a student. Professor Il-Yong Chun in our department, he is an expert on machine learning and image analysis. So he has also helped us with this project because we are definitely not experts in this area. So this is the problem. So Dr. Huang has an in vitro fertilization clinic, and they have an incubator for the embryos that is integrated with a microscope. So as the embryos are developing in the first few days of development, they can actually see and image the embryo. And what they noticed is that the embryos that grow faster tend to be the better embryos based on genetic tests that you can do before you do the embryo implantation. The embryos that grow slower tend to have some genetic abnormality, so extra chromosomes or things like that. And of course, in vitro fertilization, that process is really expensive. It's like $20,000 a procedure. You're not guaranteed a pregnancy, through this procedure, and it can be, very stressful. And also, because you don't always know which embryos are going to be viable and result in a pregnancy, sometimes multiple embryos will be transferred back into the mother, which can result in of course, like twins or triplets or things like that. But that's also a higher risk for the mother as well as for the babies. So ideally, we would want to implant, the right number of embryos for the number of babies that you want. So if you just want one baby, you would just put one embryo, and you would get the success of that process closer to 100%. So potentially measuring how fast the embryos grow is a way to do that. The problem with that is right now, these measurements have to be done manually. So there are, skilled embryologists in these clinics, but they have to look at these images and manually measure the size of the embryo for each one of these time points that you see on this graph here. So as you can imagine, this is going to be too labor intensive for most patients. So we talked to Dr. Huang about this. We decided, I think, one thing that makes a good engineer is being lazy. So not wanting to do all that work. So our solution was to make the computers do the work. And that meant doing some machine learning. So the process for that, if you're not familiar with it, is to start off with some images of embryos. Then we manually label images along with the embryologist to say, okay, this brown part that's the embryo. The blue part is the background of the image. That's not parts of the image that are not the embryo. And we do that for a lot of images and use that to train a neural network or train the computer to recognize the embryo. So all of that is a lot of work. We still haven't made it too much less work at this point. But once we do that and train the computer to analyze these images, then it becomes easy, because once we do the training and we had this set of manually labeled images, we don't need to do anything more. Now, when we get new videos and new images, we can just run it through our neural network and the computer will tell us how big the embryos are, hopefully with a good amount of accuracy. And that's what we've shown. So we're able to get more than 90% accuracy in the comparing the computer measured embryo area with the ground truth, or what the actual embryo area is. And that's visualized in these images here. So on the top row, you can just see various embryo images. The shaded green is what our neural network measured as the embryo area. And you can see that comparison to the ground truth on the bottom. So the red is what the computer program measured, and the blue outline is the ground truth of what the actual embryo is. So you can see that most of the time those curves are exactly the same, which means that we're measuring the embryo area accurately. These are just some our results on various metrics for measuring how good this measurement is compared to other types of existing neural networks. So you can, our results are in bold, so you can see we're doing pretty good. So right now we're working more with Dr. Huang on this to try to get this to help with his clinical outcomes. Switching gears again, we've also done a lot of work on reconfigurable electronics using room temperature liquid metals. This project has been, or projects related to this area have been going on for almost 10 years now in my lab, and also with some collaborations. So that's why I have so many people on this slide. So I don't have a picture of the research. So you're gonna have to wait another slide to see the research. A lot of this work has been a collaboration with Professor Shiroma, also in the EE department. We've also collaborated with Professor Host Madsen in the EE department and Professor Nasratinia at UT Dallas. And then Professor Hihara in the mechanical engineering department has also helped us on some of our work related to liquid metals. And you can see a lot of grad students have also worked on this area as well. One of the main contributors is Dr. Goff. He graduated in 2016. He's now at North Star Scientific out in Kapolei. And our most recent doctoral student graduate is Dr. Alasi, who graduated in May. He's now working at Intel. But because of COVID, he's actually still here, and eventually he'll move to Oregon. There's also many other students that have worked on this project. Some of them are Professor Shiroma's students, but I've worked closely with them on this project. And also several undergraduates, many more than I've listed here, but I've listed a few undergraduates because I kind of wanted to highlight their accomplishments. So George Zhang, you'll see his name on some of our papers with liquid metals. He graduated in 2017. He got a NSF Graduate Research Fellowship, went on to grad school at Berkeley. Is now working at Apple. Kevin Kam graduated in 2018. He did some work on liquid metals as well. And when he started his PhD program at Columbia, he also got a NSF Graduate Research Fellowship. Sasha Yamada graduated last year, and she also won a NSF Graduate Research Fellowship. She's currently at University of Illinois, Urbana-Champaign. And our most recent graduate is Nicholas Yama. He graduated in May and also got a graduate research fellowship and is going to the University of Washington PhD program. So we have a really good track record of people that have worked in this area getting these really nice NSF grad school fellowships. Other students that have also been authors on our papers as undergraduate students are Carol Kitamura. Many of you might know her. She's at Raytheon now and has done a lot of recruiting of UH students. And then Tyler Arcao has also contributed to some of our research. So the idea here is kind of along the lines of this movie, Terminator 2. For some of you, you might be too young to know this movie. So here's a clip from that movie. There is a science fiction Terminator that is made out of some kind of exotic liquid metal in the movie, and it can change its shape to match its environment. We don't want to make killer robots, but we do want to use that idea of using liquid metal that can change its shape to make a circuit adapt to a particular operating condition. So unlike regular circuits that have, they have solid metal elements in them, once you make the circuit, the metal doesn't move anymore. You can change the circuit, but you got to use something else besides the metal to change it. But we're saying, okay, well, what if you can change the metal as well to do something with that circuit while it's in operation? So this is liquid metal in our lab. This is a real video. It's not the same as the science fiction movie, but this is what we can actually do in our lab. So we can apply voltages to liquid metals and make it change its shape and revert back to its original shape as well. So we've used that to make a lot of different kinds of circuits that can change their operation after you fabricate them. Our group has kind of specialized in how to control liquid metal in various ways. So, and we focused on moving it around using some kind of electrical control, because that's going to be the easiest way to integrate it to an electrical circuit. Mechanical engineers might say, well, why don't you just pump it or something? Use hydraulic pressure. That's definitely possible, but it's a little bit less attractive to have hydraulic pumps on an electrical circuit. So we focused on using voltages. And what you're seeing here is applying some voltage across a channel, and we can get the liquid metal to move along that channel. That's by a mechanism called continuous electrowetting. We can also apply a voltage directly to liquid metal and to the end of a channel and get it to move and stretch. That's something called electrocapillary actuation. And right now, it looked like the video froze, but it didn't actually freeze. The video is still playing. We're just able to apply the voltage in such a way that we cause an electrochemical reaction that makes the liquid metal stay in place. And we've also looked at electromagnetic ways to move the liquid metal. So that's using a magnetic field and putting the liquid metal into a ferrofluid or a liquid that responds to the magnetic field, and using that to move the liquid metal. So those actuation techniques are all used to make tunable circuits. Like I mentioned before, you already have ways to make tunable circuits. Like for example, in your mobile phone, that your phone is going to switch between different frequencies of operation, and that's done by just switching in different filters in your phone. But that can be that will eventually present a limit on how small you can make a mobile device. So if you can actually move around the circuit elements, we can also make circuits that will change that way. This is a little bit more about liquid metal and its advantages as a way to make these tunable circuits. So it has a low contact resistance because it's liquid. It will fill in any irregularities at an interface. Also, because it's a liquid, if you put it in a stretchable or flexible material, you can maintain a good electrical contact as that material stretches or flexes. It's a liquid at room temperature and a little bit below room temperature, up to a pretty high boiling point. And it has a relatively high surface tension as well. So we've used that to our advantage in those actuation mechanisms that you saw. So here's an example of some circuits that we've built, or some devices that we've built using liquid metal. So here's a capacitor that we can change the capacitance over a ratio of 42:1. And these are just some configurations that we can make for that capacitor to tune its capacitance. And these can be made in flexible materials. Oops, I don't have the picture of it here, but you saw that picture earlier where, you know, there's a gloved hand holding a piece of liquid metal, and that's this tunable capacitor. We've also done a lot of work on microwave circuits and components, such as this antenna. In this case, for an antenna, what you're really trying to do is change some physical lengths of the antenna, and that will change the frequency that it operates at. So we do that in this antenna by moving the liquid metal shown in green up and down these channels. And so that changes the length of this hole in a conductor, and that changes the operating frequency of the antenna. So that's what this graph is showing here. You might not know how to read this, but so the important thing to gain from this graph is that the peak or the minimum of these curves are going to be correspond to the operating frequency or the resonant frequency of this antenna. So these different curves represent different positions of the liquid metal, and as you can see, the resonant frequency of the antenna changes. So we're effectively changing the frequency of this antenna by moving around this liquid metal. This is a different result that we change different parameters of an antenna. So this is changing the gain of the antenna, or how well it radiates its energy in a particular direction. Again, by moving around where liquid metal is on this antenna. We've also taken this idea for tunable antennas and done some work for DARPA, where we want to make an antenna that can be configured to almost any kind of shape depending upon what you want to do with that antenna. So the idea here is that if you imagine, you know, your computer screen, which you're in front of right now, it's made out of all these pixels. And as I'm showing these different photos, those pixels are turning on and off to form an image. So we could do something like that with an antenna. So imagine instead of your computer screen showing images, you are just making antennas on that computer screen. And to make an antenna element, you need conductive elements. So if we can make pixels that instead of become bright or dark, we make pixels that are filled with a conductive material or not filled with a conductive material, we can do the same kind of thing for antennas that you can do for this display. And that's our big picture motivation for this project. So the first thing we needed to do was make a pixel using liquid metal. So what we're doing here is we're moving liquid metal from below the substrate to the top of the substrate and back and forth. So this is a reversible movement. And so if the liquid metal moves in front of the substrate, that's like having the pixel on. If it moves in back of the substrate, that's like having the pixel off. And we can use one of our electro actuation methods to do this using voltages less than 10 volts. We can then, once we have a single pixel, we can make arrays of these pixels to get closer to a full 2D display of many pixels, and then we can make any antenna shape that we want. So this is a demonstration using an array along 2 axes. So you can see each one of these squares represents a liquid metal pixel. and we have a line of them in the x-axis and a line of them in the y-axis, and we can turn on different pixels to create different kinds of antenna radiation patterns. One of the applications for this would be to try to maximize the capacity of networks when you have multiple users. So this is a project that we worked on with Dr. Holst Madsen in the electrical engineering department. They have a lot of ways where if you have multiple users, you can try to minimize the effects of interference between multiple users and maximize the data transfer rates in this kind of network situation. And this can be done with the help of liquid metal antennas. So this is a simplified version of that complicated scenario on the previous slide. So here we have 2 transmitters and 2 receivers, and we want to minimize the effects of interference between these 2 pairs of transmitters and receivers. So we can do that by making these antennas reconfigurable using liquid metal. And based on some experimental work and simulation of this system, we do show that using liquid metal for those antennas and adjusting the liquid metals to help mitigate that interference can help to increase the data capacity of each one of those links. So to kind of summarize, the projects that I talked about for this presentation are the optically controlled micromanipulation system that we are using for manipulation of single cells. I've also talked about our work in different kinds of systems for analyzing single cells with not only microsystems, but with machine learning. And our various efforts in using liquid metals for reconfigurable electronics. So I'd like to thank you for your attention during this seminar. If you have any questions or any ideas of things we can work on, as you can tell, we're very open to collaborating with many different people. Thank you.
Well, thank you, Professor Ohta. I guess now the forum is open. Any questions? So I have a question. You know, for the liquid metal, right? So I guess you have to kind of encapsulize the metal, right? Otherwise, it would oxidize. Would that be a problem, or just to enclose them?
Yeah, so I didn't talk about that in detail, but the liquid metal we're using is a gallium alloy, so it does oxidize really quickly. There's a couple of ways to do that. One, like you said, is to hermetically seal it so that it's in an oxygen-free environment, but you have to get that really good. It has to be less than a part per million oxygen. Mm-hmm. And then it'll behave like a regular liquid. The other way to do it is to put it in a solution that will get rid of the oxide. So you can either do that with a really basic solution. So we use sodium hydroxide, or you can do it with an acidic solution. So other groups use hydrochloric acid, and that will also get rid of the oxide.
Thank you.
I have a question. Very nice talk. I was wondering if you could comment a little more on the fluidic physics in the first part. So do you have like thermo-Marangoni flows, or what is the physics driving the motion of the cells?
Yeah, so we have both a surface tension gradient because of that temperature gradient. And that acts on the interface of the bubble and the liquid. So that's one of the forces that's bringing the bubble towards our hotspot. And once it's over the hotspot, all the forces balance. And then that thermal Marangoni effect at that interface is also creating a convective flow surrounding the bubble as well. So in one of those videos, when we're moving around just the cell by itself, it actually looked like we're dragging it, and we were dragging it because we're using the convective flow around the bubble.
Okay, so this isn't a thin film, or there's an air-water interface there, I guess, or is it a drop, or?
Yeah, maybe let me go back a little bit. So we've done multiple kinds of cases. So normally it's in a completely enclosed chamber. So,
Okay.
we're talking about maybe about a couple of 100 microns high for the chamber, and maybe a centimeter or so in X and Y. But we've also done it in an open reservoir. So we don't have a ceiling to the chamber, because that's a little bit more friendly to a lot of biological lab procedures.
Okay, thank you.
Any other questions? If not, I guess let's thank the speaker again, and then I'll post the seminar on the web and also social media so that people can have access to. I guess this would end our seminar today, and have a great weekend, everyone. Thank you.
Thank you.
Thank you.